Thank you all for standing by and welcome to our webinar entitled Pharmaceuticals Within the Near Treatment Plants in Lake Erie's Western Basin. This webinar series called Freshwater Science will highlight Ohio Sea Grant and partnering scientists every month. Every quarter is a different focus, from human health and fish farming to harmful algal blooms and human decision making, bringing applied research to the public on issues that affect our Lake Erie communities. I am Jill Gentis-Banicki from Ohio Sea Grant and Stone Laboratory, and joining me today is Dr. Kevin McClooney from Bowling Green State University. Kevin McClooney is the Associate Professor within the Department of Biological Sciences at Bowling Green State University. His lab focuses on understanding aspects of global change in aquatic and terrestrial ecosystems. The lab integrates ecological, physiological, and isotopic approaches to understand the dynamics of populations, communities, and ecosystems, with a focus on aquatic, riparian, terrestrial, and urban environments across the U.S. We're delighted to have him here to talk about his research today. Before we do, a few things about the webinar itself. During our presentation, all participants will be in a listen-only mode. Afterwards, around 12:20, I'll conduct a question and answer session. If you have any questions, feel free to use that chat feature anytime during his talk, and I'll collect and pose your questions out to Dr. McClooney at the end of his presentation. As a reminder, this webinar has auto captioning and is being recorded to be posted onto our website for later viewing. Also, we will post a webinar survey in the chat feature toward the end of the half an hour. Please take a few minutes after the webinar to fill out that survey. It always helps us continue to bring you better webinars. So without further delay, I'd like to introduce Dr. McClooney, presenting pharmaceuticals within and near treatment plants in Lake Erie's Western Basin.
Thank you, Jill, and thank you for inviting me today. Let me share the presentation with you. Yeah, so she already read the title, so I don't need to repeat that, but I'll go to the next slide and I'll mention that I did not do this work alone, and I want to recognize my collaborators who are instrumental in this work. First, Travis Wurst in Forensic Science at BGSU, who really did pretty much all the pharmaceutical testing. Louise Stevenson, who's at Oak Ridge National Lab, and she's an ecotoxicologist, and Chris Ward in the biology department at BGSU with me, microbiologist. So I also have a number of technicians and students to thank. In particular, I want to thank Jonathan Brokaw. He's a master's student in my lab who collected most of the samples for this work. And Jeremy Canfield is a research technician in forensic science who did a lot of the wastewater chemical testing. Additionally, tons of other students involved: Alessia Saul, Emily Dwyer, Kelly Walentic, Rick Cummins, Kate Brown, Parker Caskell, and Danica Gilpin also all helped with this research. So thank you to that group. Okay, so first I want to start with our research goal. So first I want to start with the long-term research goal. Our long-term research goal is to determine the identity and effects of suites of wastewater contaminants on linked aquatic and terrestrial food webs and ecosystems. That's our long-term goal. To get there, we're doing sort of the first step of that. We need to really understand how pharmaceuticals and other wastewater contaminants vary over time and in different locations in Northwest Ohio, to be able to understand how they're affecting these food webs and ecosystems. Why does this matter? Well, one reason is understanding food web responses to real exposure scenarios, and I'll go into that a little bit more in a moment of what is typically done and the direction we're trying to head. Also, help wastewater managers better remove some of these chemicals that are sort of emerging chemicals, emerging concern that people haven't been looking at that much. And finally, it also could be used to help public health officials know patterns of potential substance abuse that they might not see via other mechanisms. So first of all, I want to talk about, how do pharmaceuticals enter streams? How do they get there? So when people take pharmaceuticals, they go into their body, they're absorbed, they're metabolized, and sometimes metabolites are produced. I'll talk about that more in a second. And either the original compound or the metabolite is then excreted. It's not the only way this can happen. Other sources, people directly flush illegal drugs, for instance, but also just prescription drugs. They can come from factories. They can also come from medical facilities. And so all of this eventually can enter the wastewater stream. Then, so that wastewater then goes to wastewater treatment plants, where there's an attempt to remove as much of various contaminants as possible. Not always thinking about pharmaceuticals specifically, but other things. And then the effluent is discharged into the stream. Sometimes these contaminants that end up in the stream can then end up in wildlife, okay? So that's sort of the connection we're looking at. Here's an example of that. This is research actually out of Australia, where they found pretty high concentrations of various pharmaceuticals in invertebrates and even spiders living along the edges of the stream. They also found them within vertebrates in the stream. So vertebrates also particularly had high concentrations of antidepressants in their tissues. So that's one that we are looking at a little more closely in our research as well. These exposures can sometimes have strong direct effects. So here's an example of a fathead minnow that was exposed to some female hormones that changed its overall phenotype and presentation and appearance. Sometimes they can be complex indirect effects. So this is an example from my own lab. This is Melanie Marshall, a former PhD student in my lab. And she looked at the effects of caffeine on biofilm. That's the sort of slimy algae stuff on the rocks on the bottom of the stream. Looked at the effects of caffeine on changing the nutrient content of that, right? So that is the base of the food web. That's what other things are eating. So if the nutrient content changes, that could have really important effects on the food web, but they're indirect, right? And what made it really complex is it actually was affected by the amount of light. So how the degree to which caffeine had an effect on biofilm nutrients depended on light. Don't have to understand the details of this graph, just basic point there. So next, I want to just mention how typical ecotoxicological research is conducted. Most studies expose model organisms to a constant concentration of a single chemical, one at a time, usually in the lab. This is very important research, but there's some problems. First of all, streams are actually a chemical soup. They have lots of different chemicals in them, right? Wastewater contaminants. This is some research that we've done in my lab, sort of outlining some of these common mixtures of chemicals that occur in streams across the US. So there is a wide suite of possible chemicals that can be found. Additionally, not only do they occur in mixtures, but they also vary over time. It makes it more complicated. This is specifically looking at some illicit drugs and how they vary over time in Europe. So you can see substantial daily variation. So back to our research goal right now. How pharmaceuticals and other wastewater contaminants vary over time and in different locations in Northwest Ohio. So I want to go through a brief summary of our methods. I'm not going into a lot of depth here. We collected wastewater influent and effluent samples from 9 treatment plants from cities of varying size between about 7,000 people and 270,000 people. We collected grab samples from all locations. So that means we just go out and we dip a pole in the water and we collect a single sample and we bring it back to the lab at a specific time point. We also collected automated samples every 3 hours from 2 treatment plants. So I'll show a picture in a second. Here it is. But automated sampler that we used to collect wastewater samples. This is that effluent samples every 3 hours. Additionally, we collected stream water samples from 3 treatment plant locations. Those were from cities that varied in size from about 13,000 to 40,000 people. Specifically, we collected samples upstream of where the effluent, the wastewater effluent comes out into the stream, near downstream, about 25 meters downstream from where that effluent comes out, and farther downstream, about 150 meters downstream. We collected composite samples each day for 1 week. So what does that mean? That means we still collected the water every 3 hours, but we combined it together into a single daily sample. But then 1 day each week, we also analyzed all of those samples separately every 3 hours. So we could see those temporal patterns. Here's an example of what this looked like. So there's effluent outfall into the stream, and then this is about 25 meters downstream, and you can see a tube coming out over here. That goes to our automated sampler that's higher up on the bank. I wanted to also mention, we did collect some other things that I'm not really going to talk about today because we don't have the measurements ready to present, but we collected biofilm samples, invertebrate samples, and emergent insect samples. So this is an emergent insect trap. We measured 19 pharmaceuticals plus PFAS. I'm not really going to talk about the PFAS today either because, again, we're not quite ready to present those results. But I am going to talk about these pharmaceuticals. And the pharmaceuticals, this is a table of them. And so you can just see each of them listed there in the table. Some of them were unregulated chemicals like caffeine, or over-the-counter chemicals like Benadryl. Or here is an unregulated, not unregulated, you need prescription, but it's not considered a substance of abuse. This is sertraline, it's an antidepressant. We'll talk a lot about that. These three were found really in highest concentrations, or sorry, most frequently detected in our samples. So these are sort of the unregulated, not considered to be illicit or over the counter. All these ones up here, though, they have they're listed on what is called a schedule, and the U.S. federal government lists drugs under the potential for abuse. And Schedule 1 and 2 are the highest potential for abuse. Schedules 3 through 5 are really much less potential for abuse. So you can see we have a variety of chemicals in here. Here's some more antidepressants that are low potential for abuse. But then we have things like fentanyl, right, which is Schedule 2, because it does have some applications medically, but it's high potential for abuse. But then I also wanted to point out we have some metabolites of these chemicals. So there's norfentanyl. That's the key metabolite of fentanyl. And this one, benzylecgonine, is the key metabolite of cocaine. That's a little bit harder to tell, but that's sort of an important one. And some of these illicit drugs or their metabolites were also detected somewhat commonly. So you can see oxycodone was detected in about 10% of our samples, whereas this metabolite of cocaine was detected in 50% of our samples. Okay, so now I want to turn your attention to geographic variation, different locations showing different patterns. It's going to be a rather complicated graph, so I want to lay it out first and show you what's going to be in there before I show you the data. The first thing I want to point out is we have our grab samples up here on the left. So everything down here is going to be grab samples. And our non-composite every 3-hour samples over here on the right. We only have that for 2 cities, City 1 and City 4. City 1 was a medium city, sampled 2 24-hour periods in June. And City 4 was a large city, sampled 1 24-hour period in March. Then we have all of our other cities listed over here, only under grab samples. I also want to note that we're protecting city anonymity here, so we're not telling you the specific locations of the cities on purpose. Other thing I want to say about this graph is there's different rows for different chemicals. So this row up top is going to be caffeine, Benadryl, the antidepressant sertraline, THC, active component in marijuana, cocaine, the metabolite of cocaine, the metabolite of fentanyl, and meth on the bottom. And the concentrations are listed over here on the left in log scale. Okay, so I want to show you some data. First, I want to mention this is a box plot. It basically shows the distribution of the data. The most of the data falls where the darkly colored box is. That's where most of the data is. But you can also see each data point there, so you can look at it that way if you want. So there's a few things you should notice. One of the things you might notice is that in this top group of chemicals that are sort of the less potential for abuse chemicals, you see and widespread use chemicals, you see a pattern where city 1 appears to always be higher than city 4. City 4 is the larger city, so there's no real reason to expect this. So this is kind of an interesting pattern. So we thought about this for a little while. This is in wastewater influent too, so it hasn't been through the treatment plant yet. And one of the things we think it might be is just seasonal differences in maybe water temperature between June and March, right? Or differences in infrastructure. What I mean by that is the network of pipes that takes the wastewater to the treatment plant, for instance, could be different amount of that and how it's affected by those seasonal differences. So we don't really think that these differences are kind of real, oh, there's higher use of caffeine in city 1 and city 4. If you go down to this group of chemicals, you can actually see the opposite pattern, where it's higher in city 4 than city 1. Given that here it was always lower and here it's higher, we think this is really probably due to differences in use, right? So what we've got is the cocaine and the metabolite of cocaine and fentanyl, the metabolite of fentanyl, much higher in city 4 than city 1. The grab samples are a little more complicated because it's just a single time point. So we just get a single data point for each of these grab samples. The main thing I want to point out is that if you look at this, you see like relatively little variation between locations for a lot of these, except for maybe Benadryl. There could be some seasonal allergy effects going on there. But then if you go down and look at this group of illicit drugs, you see a lot more variation by location, right? And so what that suggests to us is that's probably reflecting differences in use. But grab samples, there's some problems. We're going to talk about that more in a minute. Okay, so that was influent. That was water coming into the treatment plant. What about effluent? What about water going out? Are treatment plants effective at removing those chemicals? So this is a rather complicated graph again. You can see the rows are still the different chemicals. That's the same. City is on the top, city 1, sample 1, city 1, sample 2, and city 4. And then the solid lines are the influent and the dashed lines are the effluent. They're staggered on purpose because a lot of wastewater treatment plants have about 24 hours for the retention time, how long it takes to move through the plant. It varies with different factors like rainfall, but on average, that's about right. And so what that allows us to do is say, okay, this sample of water coming into the treatment plant is the same as this sample of water exiting the treatment plant. So we can look at these samples over time and see how well the treatment plant is removing those chemicals. So we can kind of note already that for caffeine, city 1 seems to be doing a pretty good job removing that chemical, right? City 4, not so much. I'll remind you that this was collected at a different time of year in March. So we'll have to think a little bit more about what's going on there in terms of why city 4 is not as effective at removing this chemical. Let's look at the other chemicals. It's a lot to take in. But if you look at it, most of the chemicals in most situations, even over here in city 4 for some of them, are greatly reduced, right? But there are some key exceptions which are interesting. Sertraline, that antidepressant that we've been talking about, is really not well removed by any of the treatment plants. Also, norfentanyl, that's the metabolite of fentanyl, where it is detected, it's not being removed very well by the treatment plants. So now we've talked about influent and effluent and treatment plant effectiveness. What about stream water? Let's look at the stream. So many of these chemicals are still present in stream water. So this is just kind of an overall view. What do we find there? We still find caffeine in the stream water, Benadryl, the cocaine metabolite, this antidepressant sertraline, and a variety of other opiates. Do they dissipate downstream? All right, so they go into the stream. What happens when they go out of the stream? So first of all, this is not, they're paired samples directly in time because the amount of time it takes to move from 25 meters downstream to 150 meters downstream is very small. So grab samples directly at the same time are comparable. So what you see, what you first notice is, right, city 1, there's almost no dissipation of that chemical downstream. City 2, maybe a little bit more, city 3 also. But in general, there's not a whole lot of dissipation as these chemicals move downstream, with some exceptions. One important thing to note is that City 1 with the least amount of dissipation is the smallest stream. City 3 is a medium stream, and City 2 is the largest stream. So there seems to be some effect of stream size, where more dissipation occurs in larger streams, which maybe isn't surprising. But despite all that, you can see differences in illicit drugs in the stream water too. So this is just looking across all of our stream water samples. And the things I want to point out is that it appears that City 1, there seems to be higher use of cocaine, right? Cocaine and the metabolite of cocaine, as well as codeine. And down here for fentanyl, it looks like higher in City 2. And down here for oxycodone, it looks like much higher in City 3. So there does appear to be that geographic variation you can see directly in the stream water as well. We talked about some variation over time a little bit already. So this really shows that you can see substantial daily variation in some streams, City 1 and City 3. You see a lot of variation in concentrations of these chemicals over time. But if you look at City 2, much less variation over time, right? And lower concentrations too, okay? And again, City 2 is the largest stream, okay? So you kind of see an effect of stream size, less daily variation in larger streams. And this is similar for hourly variation. So here's the hourly variation. I'm not going to go into a lot of detail, but much higher in City 1, which has the smallest stream compared to the largest stream. So in summary, what have we found? We found substantial geographic variation in controlled substances. We found that most substances are greatly reduced by most treatment plants most of the time. Not always, though. Most substances don't dissipate downstream, but of course that effect is determined by the size of the treatment of the stream as well. Substantial variation over time. And again, the size of the receiving water body greatly affects concentrations and their variation over time, which, brings up this old adage, the solution to pollution is dilution, right? So that does still seem to apply here. Another way to summarize is thinking back to food web exposure. Some chemicals, there is more constant exposure. Benadryl, sertraline appeared to be a little more constant over time. But more pulsed for some chemicals, like some of these illicit drugs, depending especially on stream size, though. And we did find evidence that there were different chemical mixtures for different cities, right? And this seems to be related to differences in use and differences in treatment, potentially as well. And a final point I want to make is that the GRAB in composite samples do have some limitations to their value due to all that temporal variation we saw. But they still have some value. So I don't think we want to just say, oh, they don't matter. I think there's still important sort of ways to look at the system if that's all the resources you have to invest. So what are some research needs? Well, now that we know a little bit more about which chemicals are more constant and which ones are more pulsed, we can start looking at the effects of these pulsed chemical exposures as well as mixed exposures on aquatic life and food webs. One other thing we want to do is we want to get a better understanding of the effects of differences in infrastructure in cities, seasonal differences, and treatment effectiveness differences, and how that really changes these concentrations. And then finally, it's something that we'd like to head in the direction of. We're wondering if we can make GRAB samples more useful by modeling that temporal variation that we see. So look at the temporal variation first, model that, and then place the GRAB samples within that context. So with that, that's the end of my presentation. I just want to say thank you all for listening, and happy to take any questions.
All right, great. We have gotten quite a few questions. So let's see. I will ask Dr. McClooney as many as we can here. Let me see. First question we had was, it was kind of early on in your presentation. There's a question of, was there a consideration given to potential interactions or effects that the pharmaceuticals have on each other in this study?
Yeah, not in this study, because we're mostly focusing in on just seeing where those chemicals are found and when, rather than looking at, if you're talking about interactions on how they affect organisms, that's sort of a future direction. And a number of people are thinking about this now, and people are starting to test the effects of chemical mixtures, but it's complicated, right? There's a lot of chemicals, and how do you do that? Do you expose the organisms to all of the chemicals that they might encounter? Or like one at a time, or two at a time, or three at a time, right? So there's a huge number of potential combinations, right? So that makes it challenging.
Do you have a feel for the percent of stream flow contributed to wastewater treatment plant effluent for these streams?
Well, I think maybe this was maybe earlier in my presentation. So I sort of talked about how that some of there was a clear effect of the size of the receiving stream on how you saw that temporal variation over time, as well as the concentration. We haven't worked that up explicitly yet, and we were only able to really sample 3 streams for this particular project. So that's kind of a future direction to get a better handle on that. We'll get a rough idea from this project, but we need to look at a larger number of streams to really develop that relationship between, what's being coming out in the effluent versus what is already in the stream, and how that influences things.
Okay. Your data shows a reduction of the tested chemicals influent versus effluent. Are you assuming the chemicals just get entrained in the sludge, or are they actually being digested? And there's quotes there. If entrained in the sludge, will this impair beneficial use of the sludge?
That's a great question, and we haven't started looking at sludge at all. I think others have done some work on that topic. I think it's going to depend a lot on the chemical, a whole lot on which chemical it is. Some of those chemicals, like cocaine, break down relatively easily, but they break down into those metabolites, right, that are much more resilient and don't break down as easily. So it's going to depend highly on the chemicals. Some are more recalcitrant and don't break down very easily. And some of that may end up in the sludge. Yep.
Okay. We had a couple questions because there were a couple people that came on a little late, and they were asking, could you clarify which sites what your research, what areas this research was taking place, what city sites?
Yeah, no, we're trying to protect city anonymity.
Oh.
It's the promise we made to the wastewater treatment plants, and I think it's good there's a variety of social implications. So we're not going to report what cities were involved.
Okay.
But it is Northwest Ohio. That's all I can say.
Okay. Within the study time frame, did any wastewater treatment bypass events occur? And if so, did sampling occur?
That is a really good question. We haven't dived back to see if there were any reported bypass events. We do have some suspicions. I don't know. I could show you a graph, but I'll probably just talk about it. In one of the cities, there appears to be a big peak on all the chemicals that we measured, and we know it coincided with a flood, because we actually had to stop sampling right after that and restart later. Because of that flood event. So it does appear like there might have been something going on there at the beginning of that flood.
All right.
We can't say for sure.
Okay. For the stream sampling, did you sample upstream and downstream of the effluent to evaluate the change in concentration?
Yes, we did. And I can show a graph of that, but I think I'll just say we did. And for the most part, it's low, except for one of the cities that discharged into the smallest water body, or one of the smallest water bodies. We believe that we had some upstream readings that were actually backflow, where the wastewater effluent went upstream. So, but except for that particular instance, yeah, most of these chemicals were much lower upstream or not present at all.
Okay. We got a couple of questions asking what is the current regulatory requirement for wastewater treatment plant discharge for pharmaceuticals?
I don't know the answer to that question, so I'll say that up front. But I'm pretty sure that that most pharmaceuticals are not regulated for wastewater treatment. There may be a few that are, but I think most of them are not currently regulated or tested for.
Okay. Could you talk a little bit about your extraction and sampling methods?
Yes, and for that, I will share a slide because I want to make sure I get this right. And you know, that is some of that is not my expertise. Some of that is expertise of the forensic science group, right? So I want to make sure that I talk about it correctly. We stored samples on ice, including in the auto sampler when we collected them that way. Then we froze samples after collection. Then for the influent samples, we did Anton-Par microwave extraction, where we had done some work to test temperature and duration of that to optimize that correctly. Followed by solid phase extraction of all the samples, whether they were influent, effluent, stream water. Different amounts of influent and effluent, which affected our detection limits. You might have seen our detection limits were different in some of the slides above. And our solid phase extraction used these hydrophilic, lipophilic balanced solid phase extraction columns. Then they were dried down, then dissolved in 1 milliliter methanol. And then we used LCMS with appropriate standards and calibration curves. And I don't know if I'll go all into all this, but you know, I don't know who the question is from, but yeah, if you have a high level of understanding, then you probably want to read some of this other stuff as well.
All right. Another question we got was the detection. They were asking about the detection of THC in some of the samples. Do we know if they are in any way scrutinized in Ohio, for example, Ohio EPA or the Department of Health?
Well, yeah, I mean, I don't think that this kind of wastewater detection of illicit drugs is being used in Ohio very much, if at all. There's, to my knowledge, there's only a handful of studies doing this sort of thing across the entire United States. So I'd be surprised if it's a regular thing. I'm not aware of something where it's a regular thing. I do think that it could be useful information. I'll stop sharing. Could be useful information for public health, right? To think about, you know, where do we have particular problems? And, you know, you might say, oh, well, we'll know from police records, which might be true, but there might be particular neighborhoods where police are not catching what's going on, right? And so you may have substance abuse problems in locations that you don't realize, for instance.
Right. Are there any of the streams that you tested used as drinking water sources? And if so, have you tested to determine if conventional drinking water facilities can remove them, or do they pass through?
Well, in some sense, since it's Northwest Ohio, like all of them eventually flow into Lake Erie, right? So definitely at some point along the way, there is some kind of water intake that some city is using. But it could be quite a bit downstream. I'm not aware of any intakes like near downstream of these effluent outfalls. So, you know, that we have not looked at yet. What are the concentrations of these chemicals coming into drinking water treatment? So that's a good question. I suspect they'll be pretty low. You know, we're really sort of focusing more on the stream food webs. But, you know, some of the more recalcitrant ones that don't seem to dissipate very much, like sertraline, for instance, might be good to look at that in intake water.
We've gotten several questions that might be a little bit out of your domain, but I'll ask and then you tell me. Are there standard methods that US EPA approves for these chemicals in wastewater and streams? If so, does this sampling meet those standards?
Some, but not all. So I think that, you know, when you're looking at contaminants of emerging concern, they don't always have methods developed for them. And so, you know, we looked at those methods. We are not necessarily following those methods perfectly, but we have a lot of similarities with those EPA methods. You know, obviously they're not for all chemicals. So for the ones that were relevant.
All right. we had quite a few people asking about journal articles. If there are any journal articles right now of your research. That
Yeah,
was the first question.
Not of this research. I was literally doing the data analysis and graphs over the weekend, so it's hot off the presses. So this research has not been published yet. I did mention a couple of papers that have been published with my former PhD student, looking at suites of wastewater, well, not just wastewater contaminants, but suites of contaminants in streams around the US, as well as the work we've done looking at the effects of caffeine on biofilms and food webs and streams.
Another question that we had dealing with journal articles. First, one person was asking if you were familiar with or have done any work with the Procter and Gamble's research that they're working on a study of combined pass-through materials.
No, I am completely unfamiliar with that. So that's something I should look into, I guess. I haven't heard about that at all.
And another question in relation to this, are there any journal articles or publications that you would recommend for people to take a look at if they wanted more information?
Yeah. I had a slide that I actually took out. Let's see, maybe I'll just share screen real quick because that'll be easier than me trying to remember. So this one, Bernhardt et al. 2017, I don't have the full reference, but this is a really good study that talks about pharmaceuticals in stream water, in our water bodies. And it's kind of a review article. So that would be a good place to start. And there's been a variety of studies, handful of studies starting to look at these, how they're affecting biofilms, how they're found in different animals. So there's some emerging research on this, but there's still a lot more to do. But this would be a good place to start.
All right. I was just trying to see, there were a couple of questions that we may follow up later. Let me see. I'm like looking at if there's any other ones that the major ones we have taken care of. I think that those are the major ones. There were a couple that we may follow up with later, just from a time perspective. So let me just close here because we are actually a little past. So let me see. I wanted to thank you, Dr. McClooney, for your willingness to talk with us today about your research. It was really an excellent discussion. I also wanted to remind everyone that our survey URL for the webinar is in the chat feature, so please take a few minutes to fill that out. This webinar series is sponsored by Ohio Sea Grant and will continue in August with Ohio State University's Jim Hood, who will be talking about his research on phosphorus cycling in agricultural streams in the Maumee River watershed. The registration link is in the chat. Thank you again, Dr. McClooney, and all the participants on this webinar. We hope that this was beneficial and hope you'll join us again in an upcoming webinar. Thank you again, Dr. McClooney. This was really, really interesting. We really appreciate it.
Thanks, Jeff.
Thank you.